Good afternoon, students. Today we are having our next lecture, which is general aspects of physiology of endocrine system and hypothalamus and pituitary gland functions. So today we will be discussing hormones, receptors and target cells action, factors affecting the concentration of hormone, mechanism of action of hormones on target cells, and we will discuss all hormones that are secreted from hypothalamus and pituitary gland. So, endocrine system is composed of endocrine glands, which are ductless glands secreting chemical messengers into the circulatory system. There is a difference between exocrine glands and endocrine glands. That endocrine glands, they have ducts and they carry the secretions to the surfaces, while endocrine glands, they secrete hormones directly into the bloodstream, and these chemical messengers are called hormones. In addition to the classical endocrine hormones, many other cells in the body secrete hormones. For example, myocytes in the atria of the heart and scattered epithelial cells in the stomach and small intestine can be considered sometimes as diffuse endocrine system. Here you have a table with the list of all hormones that are secreted in our body, the target cells and the main functions. We will be discussing in details about each of them during our today's lecture and next week lecture. So, what are hormones? Hormones are chemical messengers secreted into the blood or extracellular fluid by one cell that affect the functioning of other cells. Most hormones circulate in blood, coming into contact with essentially all cells. However, a given hormone usually affects only a limited number of cells, which are called target cells. A target cell responds to a hormone because it bears receptors for the hormone. So you can look at this picture that different types of hormones have its own special particular receptor, and this hormone can be bind only to this one particular receptor. It cannot bind to any receptor. So hormone receptors are found either exposed on the surface of the cell or within the cell, depending on the type of hormone. In very basic terms, binding of hormone to receptor triggers a cascade of reactions within the cell that affects function. There are different types of hormones actions on target cells, and we can classify all these actions in three types. So first one is endocrine action, when hormone is distributed in blood and binds to distant target cells. Second one is called paracrine action, when hormone acts locally by diffusing from its source to target cells in the neighborhood. And third one is autocrine action. The hormone acts on the same cell that produces it. So here you have a picture. Autocrine, the feature is that the hormone can be synthesized to distant target cells, affect distant target cells. Paracrine, hormone can affect only neighboring cells. And autocrine, hormone can affect only same cell that's secreted. And only hormones that have endocrine action can be considered as true hormones. Other hormones that are having paracrine or autocrine action are called as tissue hormones. All hormones can be classified in 4 groups depending on the chemical structure and the properties. So these groups are peptides and proteins, steroids, amino acid derivatives, and fatty acid derivatives, eicosanoids. So let's talk about each of these groups. Peptide and protein hormones, they vary considerably in size and post-translational modifications, ranging from peptides as short as 3 amino acids to large multi-subunit glycoproteins. Many protein hormones are synthesized as prohormones or precursors. Then they proteolytically clipped to generate the mature form. In other cases, the hormone is originally embedded within the sequence of a larger precursor, then released by multiple proteolytic cleavages. Peptide hormones are synthesized in endoplasmic reticulum, then transferred to the Golgi apparatus and packaged into these secretory vesicles for export. They can be secreted by one of the 2 pathways. First one is regulated secretion. It means that cell stores hormone in secretory granules and release them in burst when stimulated. This is the most commonly used pathway and allows cells to secrete a large amount of hormone over a short period of time. And the second type is constitutive secretion. The cell does not store hormone, but secretes it from secretory vesicles as it is synthesized. Most peptide hormones circulate unbound to other proteins, but exceptions exist. For example, insulin-like growth factor 1 binds to one of several binding hormones, and in general, the half-life of circulating peptide hormones is only a few minutes. Steroids. They are lipids in nature and are derivatives of cholesterol. Examples are sex steroids such as testosterone and adrenal steroids such as cortisol. They are synthesized from cholesterol, which is converted into pregnenolone. Pregnenolone is formed on the inner membrane of mitochondria, then shuttled back and forth between mitochondrion and the endoplasmatic reticulum for further enzymatic transformations involved in synthesis of derivative steroid hormones. Newly synthesized steroid hormones are rapidly secreted from the cell with little of any storage. Increases in secretion reflects accelerated rates of synthesis. Following secretion, all steroids bind to some extent to plasma proteins. This binding is often low affinity and non-specific, and mostly steroids are bound to albumins. But some steroids are transported by specific binding proteins, which clearly affects the half-life and rate of elimination. After steroids performed the action, they are eliminated by inactivating metabolic transformation and excreted by urine or bile. Amino acid derivatives. There are tyrosine and tryptophan and glutamic, examples of amino acid derivatives. And what are these hormones that are synthesized from these amino acid derivatives? So thyroid hormones, which are T3 and T4, they are synthesized from amino acid tyrosine, as well as catecholamines, which include epinephrine and norepinephrine. The circulating half-life of thyroid hormones is on the order of a few days. They are inactivated primarily by intracellular deiodinases, Catecholamines, on the other hand, are rapidly degraded with circulating half-lives of only a few minutes. Tryptophan, another hormone, another amino acid derivative, which is a pre-hormone for serotonin and the pineal hormone melatonin, and histamine, which is synthesized from glutamic acid. Fatty acid derivatives, next group of hormones. Arachidonic acid is the most abundant precursor for this group of hormones. Stores of arachidonic acid are present in membrane lipids and released through the action of various lipases. The specific eicosanoids synthesized by a cell are dictated by the battery of processing enzymes expressed in that cell. Eicosanoids are a large group of molecules derived from polyunsaturated fatty acid. The principal groups of hormones of this group are prostaglandins, prostacyclins, leukotrienes, and thromboxanes. These hormones are rapidly inactivated by being metabolized and are typically active for only a few seconds. What are the factors that can change the level of secretion of hormones or amount of concentration of hormones? So first one is rate of production. Synthesis and secretion of hormones are the most highly regulated aspects of endocrine control. Such control is mediated by positive and negative feedback loops. Rate of delivery is the second factor. An example of this effect is blood flow to a target organ or group of target cells. Obviously, that high blood flow delivers more hormone than low blood flow. Third factor is rate of degradation and elimination. Hormones, like all biomolecules, have characteristic rates of decay and are metabolized and excreted from the body through several routes. Shutting off secretion of a hormone that has a very short half-life causes circulating hormone concentration to plummet, but if a hormone's biological half-life is long, effective concentration persists for some time after secretion ceases. Now let's talk about how level of hormone can be regulated. So there are two types of feedbacks, positive and negative. Negative feedback is seen when the output of a pathway inhibits input the pathway. In simple words, when one hormone level is low, the second one, which is releasing hormone, is increased to maintain the low level of one hormone to bring back this low level of first hormone to the basic level. To better understand this negative feedback, let's explain this by the help of thyroid hormone secretion. It's a classical example of negative feedback loop. So we have thyroid releasing hormone, which is synthesized in the hypothalamus. Then we have a pituitary gland, which releases thyroid stimulating hormone, and we have thyroid gland, which releases thyroid hormones T3 and T4. So how negative feedback loop is working? So if level of T3 and T4 is low, then it's a stimulatory signal for neurons in hypothalamus to release more of TRH. If TRH start to synthesize in great amount, then it's affect pituitary gland in stimulatory way to release more of TSH, thyroid stimulating hormone. If we have more greater amount of TSH, then it is a positive, it's a stimulatory effect of thyroid gland to release more T3 and T4. So the results bringing back of thyroid hormones level to normal. So once again, in simple words, that decreasing of one hormone leads to increasing of second one, releasing hormone. And positive feedback occurs when we have both hormones level increasing. So if, for example, thyroid hormones level increase, then it leads a stimulatory effect to release of more TRH. So it's a positive feedback when both of hormones level of synthesizing increase. But most of the hormones in our body are regulated by the help of negative feedback. So one hormone is low, then releasing hormone that is released from hypothalamus is increased. But you also have to understand that level of each hormone is changing during the day, during the night, during the week, during the months. So here you have example of luteinizing hormone. Here you have a graph of synthesing of concentration of luteinizing hormone in blood of a female dog of a period of 8 hours, with samples collecting every 15 minutes. So you see that level of luteinizing hormone is not same during these 8 hours, and such a way of synthesizing of hormone is called pulsatile nature. And some of hormones, like in case of luteinizing hormones, level are changed depending on the stages of reproductive cycle. And some other hormones are changing during the day, means that in morning its level is higher than in the evening, and some hormones is vice versa. In the morning the level is low, but in the evening this level is high. So you have to understand that level of each hormone is not same all the time. And for testing the level of hormone, of any hormones, you have to test patient only in early morning in case of not eating for at least 8 hours before. So how do hormones change the target cells? Hormones, as I said previously, are chemical messengers that invoke profound changes within target cells. So it can be accomplished by two ways. First one is activation of enzymes and other dynamic molecules. Most enzymes shuttle between conformational states that are catalytically active versus inactive. Many hormones affect the target cells by inducing such transitions, usually causing an activation of one or more enzymes. Because enzymes are catalytic and often serve to activate additional enzymes, a seemingly small change induced by hormone receptor binding can lead to widespread consequences within the cell. And second way is modulation of gene expression. Stimulating transcription of a group of genes clearly can alter a cell's phenotype by leading to a burst of synthesis of new proteins. Similarly, if transcription of a group of previously active genes is shut off, the corresponding proteins will soon disappear from the cell. So as I said previously, there are two types of receptors. First one is called cell surface receptors. In this case, a receptor is present on the plasma membrane. And the second one is intracellular receptors. In this case, receptor is localized in the cytoplasm or in the nucleus. And what are the groups of hormones which are used cell surface receptors? These are proteins and peptides, catecholamines and eicosanoids. And only steroids and thyroid hormones are using intracellular type of receptors. And mechanism of action of these two types of hormones is different. So what is the mechanism of action in case of cell surface receptors? So first step, the water-soluble hormone is bound to its receptor and the G protein is activated. Second step, the activated alpha subunit with GTP bound to it binds to and activates an adenylate cyclase enzyme so that it converts ATP to cyclic AMP and to inorganic phosphatases. Third step, the cyclic AMP activates protein kinase enzymes, which phosphorylate specific enzymes activating them. The chemical reactions catalyzed by the activated enzymes produce the cell's response. And the last step, phosphodiesterase enzymes activate cyclic AMP by converting cyclic AMP to AMP. Now let's talk about the mechanism of action of intracellular receptors. So first step that most hydrophobic steroids are bound to plasma protein carriers. Only unbound hormones can diffuse into the target cell. Steroid hormone receptors are in the cytoplasm or nucleus. One second. Some steroid hormones also bind to membrane receptors that use second messenger system to create rapid cellular responses. Then the receptor hormones complex binds to DNA and activates or represses one or more genes. Activated genes create new mRNA that moves back to the cytoplasm, and translation produces new proteins for cell processes. Okay, now let's move to the next subtopic of today's lecture, which is hypothalamic-pituitary system. So, pituitary gland is very important structure in our body since it's synthesizes a battery of hormones, but the highest level of endocrine control is associated with hypothalamus. So hypothalamus can release two types or two groups of hormones which can affect pituitary gland. So first group is releasing hormones that can stimulate pituitary gland, and second one is inhibitory hormones, which can obviously inhibit pituitary gland. So now let's discuss about each hormone that is synthesized from pituitary gland, its effects and the ways of controlling. So first one is growth hormone or somatotropin. So growth hormone is considered as protein hormone. It is secreted by these cells called somatotrophs in the anterior pituitary. It is a major participant in control of several complex physiologic processes, including growth and metabolism. Growth hormone is also of considerable interest as a drug used in both humans and animals, and it has two types of effects. First one is direct effects, which are the results of growth hormone binding its receptors on target cells. Fat cells, for example, have growth hormone receptors, and growth hormone stimulates them to break down triglycerides and suppresses the ability to take up and accumulate circulating lipids. And second is indirect effects that are mediated primarily by insulin-like growth factor 1, a hormone that is secreted from the liver and other tissues in response to growth hormone. A majority of the growth-promoting effects of growth hormone is actually due to insulin-like growth factor 1 acting on its target cells. So here you have a picture representing these indirect and direct effects. So the indirect effects is represented by stimulating proliferation of chondrocytes, resulting in bone growth, as well as affecting muscles and adipose tissues by enhancing protein synthesis and cell proliferation. And indirect effect is represented by enhancing lipolysis and increasing blood sugar level. So growth hormone affect all types of metabolism, protein metabolism, fat metabolism, and carbohydrate metabolism. So in general, growth hormone stimulates protein anabolism in many tissues. This effect reflects increased amino acid uptake, increased protein synthesis, and decreased oxidation of proteins. Second one is fat metabolism. Growth hormone enhances the utilization of fat by stimulating triglyceride breakdown and oxidation in adipocytes. Growth hormone is also one of the battery hormones that serves to maintain blood glucose within a normal range. Growth hormone is often said to have anti-insulin activity because it suppresses the abilities of insulin to stimulate uptake of glucose in peripheral tissues and enhance glucose synthesis in the liver. How level of growth hormone secretion can be controlled? So what are the stimulatory factors? First one is growth hormone releasing hormone that is released from hypothalamus, which can stimulate anterior pituitary to synthesize more growth hormone. Second one is ghrelin released from stomach, which can also stimulate anterior pituitary. And somatostatin, which is released from hypothalamus, but has inhibitory effect on anterior pituitary that results in decreasing synthesizing of growth hormone. Also, the growth hormone secretion is the part of negative feedback by the help of insulin-like growth factor 1 that is released from liver. So high blood levels of IGF-1 lead to decreased secretion of growth hormone, not only by directly suppressing the somatotroph, but by stimulating release of somatostatin from the hypothalamus. So what are the diseases associated with low and high level of growth hormone? So first one is dwarfism. It's a deficiency in growth hormone or defects in its binding to receptor, as seen as growth retardation or dwarfism. The manifestation of growth hormone deficiency depends upon the age of onset of the disorder and can result from either heritable or acquired disease. Next disease related to excessive growth hormone secretion is called giantism. The onset of this disease is in young children or adolescents. It is a very rare disorder, usually resulting from a tumor of somatotrophs. And you can see the picture. On the left, the man is of a normal height, average height. The second man with the giantism disease, and the third one with dwarfism. But there is another one disease which is called acromegaly that results from excessive secretion of growth hormones, but the onset of this disease compared to giantism usually in adults, and the reason for this disease usually is benign pituitary tumor. Clinical signs of acromegaly include overgrowth of extremities, soft tissue swelling, abnormalities in jaw structure, and cardiac disease. The excessive growth hormone and IGF-1 also lead to a number of metabolic derangements, including hyperglycemia. So here you have a picture of a woman who suffers from acromegaly. If you look at the first two pictures, when she was a little girl and young woman, she didn't have any symptoms of acromegaly, but in her adult ages, her jaw start to overgrow, her fingers and hands start to also overgrow. So it's one of the differences between giantism and acromegaly, that in case of giantism, there is proportional overgrowth of all body parts. And in case of acromegaly, only some bones are overgrowth, like in this case, jaw and hands are bigger than other body parts compared to average one. Next hormone is thyroid stimulating hormone, which is also known as thyrotropin, secreted from the anterior pituitary cells called thyrotrophs. Finds its receptors on epithelial cells in the thyroid gland and stimulates that gland to synthesize and release thyroid hormones. Thyroid stimulating hormone is a glycoprotein hormone composed of two subunits which are non-covalently bound to one another. And the alpha subunit of thyroid stimulating hormone is also present in two other pituitary glycoprotein hormones, such as luteinizing hormone and follicle stimulating hormone. The most important controller of thyroid stimulating hormone secretion is thyroid releasing hormone secreted in hypothalamus. It's a classical example of negative feedback circle, which we were discussing a few minutes ago. Next one is adrenocorticotropic hormone or corticotropin, which main effect is to stimulate adrenal cortex. It stimulates secretion of glucocorticoids more specifically, such as cortisol, and has only little control over secretion of aldosterone, the other major steroid hormone from the adrenal cortex. So adrenocorticotropic hormone is secreted from the anterior pituitary in response to corticotropic releasing hormone from hypothalamus. Corticotropin releasing hormone is secreted in response to many types of stress, which makes sense in view of the stress management functions of glucocorticoids. And it is also example of negative feedback. So corticotropin releasing hormone can stimulate or inhibit synthesis of adrenocorticotropic hormone from pituitary gland by the help of level of cortisols. So as I said previously, all hormones are synthesized from pre-hormones or precursors. And precursor for adrenocorticotropic hormone is pro-opiomelanocortin, which is also called as Big Mama. And many other hormones are synthesized from this big precursor by proteolytical changes. And such hormones are also lipoproteins, endorphins, and enkephalins. Next hormone, which is synthesized from pituitary gland, is prolactin. Prolactin is a single-chain protein hormone closely related to growth hormone. It is secreted by so-called lactotrophs in the anterior pituitary. It is also synthesized and secreted by a broad range of other cells in the body, most prominently various immune cells, the brain, and the decidua of the pregnant uterus. Prolactin is synthesized as a prohormone, as many other hormones, and its main physiological effects are mammary gland development, milk production, and it also has some effect on immune function. So mammary gland development function. Prolactin induces lobulo-alveolar growth of the mammary gland. Alveoli are the clusters of cells in the mammary gland that actually secrete milk. And prolactin stimulates lactogenesis, which is milk production, after giving birth. Prolactin, along with cortisol and insulin, act together to stimulate transcription of genes that encode milk production. The prolactin receptor is widely expressed by immune cells, and some types of lymphocytes synthesize and secrete prolactin. So these observations suggest that prolactin may act as an autocrine or paracrine modulator of immune activity. How level of prolactin secretion is regulated? Most of the hormones that are secreted by pituitary gland are controlled by hypothalamus in stimulatory way. So mainly hypothalamus stimulate pituitary gland to release its hormones, but prolactin is exception because hypothalamus tonically suppresses prolactin secretion from the pituitary gland. So if we cut connection between hypothalamus and pituitary gland, the level of prolactin will increase, but level of all other hormones that are released from pituitary gland will dramatically fall. So just remember that prolactin is the only one hormone that is suppressed by hypothalamus. Next hormone which can inhibit prolactin secretion is dopamine. It has inhibitory effect. Dopamine is secreted into portal blood by hypothalamic neurons, binds to receptors on lactotrophs, and inhibits both synthesis and secretion of prolactin. Another factor that can affect prolactin secretion is level of estrogens. So gonadotropin releasing hormones and thyroid releasing hormones, they can also affect level of prolactin secretion from pituitary gland. Next two types of hormones are luteinizing and follicle stimulating hormones, which are considered as gonadotropins. These hormones are not essential for life, but they are important for reproductive function. So let's discuss the effects. So luteinizing hormones stimulate secretion of sex hormones in both males and females. In testes, luteinizing hormone binds to receptors on Leydig cells, stimulating synthesis and secretion of testosterone. TECA cells in the ovary respond to luteinizing hormone stimulation by secretion of testosterone, which is converted into estrogen by adjacent granulosa cells. In females, ovulation of mature follicles on the ovary is induced by a large burst of luteinizing secretion known as the pre-ovulatory luteinizing hormone surge. Residual cells within ovulated follicles proliferate to form corpora lutea or corpus luteum, which secrete the steroid hormones progesterone and estradiol. Progesterone is necessary for maintenance of pregnancy, and in most mammals is required for continued development and functions of corpus luteum. The name luteinizing hormone derives from this effect. Follicle stimulating hormone stimulates the maturation of ovarian follicles, and administration of follicle stimulating hormones to humans and animals induces superovulation or development of more than the usual number of mature follicles, and hence an increased number of mature gametes. Follicle stimulating hormone is also critical for sperm production. It supports the function of Sertoli cells, which in turn supports many aspects of sperm cell maturation. Regulation of gonadotropins secretion is regulated by the help of negative feedback. So if testes release great amount of testosterone, it's a signal for neurons of hypothalamus to release less of gonadotropin releasing hormone, which affect pituitary gland, which result in less concentration of luteinizing, less synthesizing of luteinizing and follicle stimulating hormone. Same with ovaries. If level of estrogens and progesterones is low, then hypothalamus start to synthesize more gonadotropin releasing hormone, and the result is a stimulatory effect on pituitary gland to release more luteinizing and follicle stimulating hormone. By knowing the effects of these two hormones and mechanism of the controlling of the concentration in our blood, we can use it in pharmacology. So we can use this in oral contraception. As we discussed above, progesterone and estrogen inhibit luteinizing hormone secretion, and these oral contraceptives are effective because they inhibit luteinizing hormone, and the result, ovulation is suppressed. And if there is no ovulation, then there is no pregnancy. So this is the general idea of oral contraception, is to inhibit ovulation by the help of inhibiting of luteinizing hormone. Next hormone, which is synthesized in pituitary gland, is antidiuretic hormone or vasopressin. Its effects the target cells for antidiuretic hormone is collecting ducts of the kidney, and the results is increased reabsorption of water back into the circulation. So the main effect of antidiuretic hormone is to decrease urine release by increasing reabsorption of water, which leads to increase in blood volume. And also, antidiuretic hormone has some effect on vascular system. So in many species, high concentration of antidiuretic hormone causes widespread constriction of arterioles, which leads to increased arterial pressure. How level of antidiuretic hormone is controlled? So if you look at the graph on the right, you will see that plasma osmolarity is the most potent effect for synthesis of antidiuretic hormone. So osmoreceptors are the receptors that can sense level of osmolarity in our body. So if plasma osmolarity increases, then plasma concentration of antidiuretic hormone increase as well. And when plasma osmolarity is below a certain threshold, the osmoreceptors are not activated and secretion of antidiuretic hormone is suppressed. Secretion of antidiuretic hormone is also stimulated by decreases in blood pressure and blood volume, conditions sensed by stretch receptors in the heart and large arteries. Changes in blood pressure and volume are not nearly as sensitive a stimulator as increased osmolarity, but are also potent in severe conditions. Also, nausea and vomiting can be stimulatory factors for releasing of antidiuretic hormone. Oxytocin, our last hormone for today. It is a peptide hormone, which is synthesized in hypothalamic neurons and transported down axons of the posterior pituitary for secretion into blood. Oxytocin is also secreted within the brain and from a few other tissues, including the ovaries and testes. Oxytocin differs from antidiuretic hormone in 2 of the 9 amino acids, and both hormones are packaged into granules and secreted along with carrier proteins called neurophysins. So what are the effects of oxytocin? 3 main physiological effects are stimulation of milk ejection, stimulation of uterine smooth muscle contraction at birth, and establishment of maternal behavior. So stimulation of milk ejection. Milk is initially secreted into small sacs within the mammary gland called alveoli, from which it must be ejected for consumption or harvesting. Mammary alveoli are surrounded by smooth muscle cells, which are prominent target cells for oxytocin. Oxytocin stimulates contraction of myoepithelial cells, causing milk to be ejected into the ducts and cisterns. Do not be mistaken with the effects of oxytocin and prolactin. So oxytocin is responsible for only ejection of milk, while prolactin is responsible for synthesis of milk. So two different processes, synthesis of milk and ejection of milk. And two different hormones are regulating these processes. Okay, next physiological effect of oxytocin. That at the end of gestation, the uterus must contract vigorously and for a prolonged period of time in order to deliver the fetus. During the later stages of gestation, there is an increase in abundance of oxytocin receptors on uterine smooth muscle cells, which is associated with increased irritability of the uterus. Oxytocin is released during labor when the fetus stimulates the cervix and vagina, and it enhances contraction of uterine smooth muscle to facilitate parturition or birth. And establishment of maternal behavior, another physiological effect of oxytocin. Successful reproduction in mammals demands that mothers become attached to and nourish the offspring immediately after birth. It is also important that non-lactating females do not manifest such nurturing behavior. The same events that affect the uterus and mammary gland at the same time of birth also affect the brain. So during parturition, there is an increase in concentration of oxytocin in cerebrospinal fluid, and oxytocin acting within the brain plays a major role in establishing maternal behavior. So what can affect the level of oxytocin secretion? So the most important stimulus for release of oxytocin is physical stimulation of nipples. The act of nursing or suckling is relayed within a few milliseconds to brain via a spinal reflex arc. These signals impinge on oxytocin secreting neurons, leading to release of oxytocin. Another numbers of factors that can inhibit oxytocin release are acute stress, because in case of acute stress, there is a burst of catecholamines, which released from adrenal gland and which can suppress oxytocin secretion. And also both the production of oxytocin and response to oxytocin are modulated by circulating levels of sex steroids. So the burst of oxytocin released at birth seems to be triggered in part by cervical and vaginal stimulation by the fetus, but also because of abruptly declining concentrations of progesterone. Another well-studied effect of steroid hormones is the marked increase in synthesis of uterine oxytocin receptors late in gestation resulting from increasing concentrations of circulating estrogen. Okay, this is all for today's lecture. See you next week. Have a nice day and bye-bye.